the fragments of basal fungus comb, which has differentiated into very hard type of structure, forming a basal disc, but to the basal disc is attached very hard this crustose fungus comb, where we do not see any presence of the anamorphic or asexual stage of this Termitomyces species, that is sporodocial form. So this is a totally different type of structure, and we really do not know what is it that makes this structure and what cements it and gives it this very crustose, rock hard, like cartoon like consistency and structure. And from this is almost dry, and from this it is very interesting to see that unlike any solid state fermentation process that we see in industrial operations, the transition of the mass transfer from this fungus garden, that is the comb, to a living species occurs from this point. So this is a very important point in terms of emergence of the species, and I believe that among all the Basidiomycetes known, that species of Termitomyces which are only found obligately with the termites, they have an extremely complex life cycle. Their entire existence depends on this stage, and then the transition, and then emergence. And after emergence also it faces a lot of issues like rain and the attack by snails and beetles, and considering all those factors, the basidiospore discharge and dispersal is going to be a very interesting phenomenon, because the life cycle of this species seems to be intimately associated with the needs of the fungus cultivator termites to recharge this material, because this is now almost discarded, means this is not under active consumption. So this fungus comb looks to be a part of a pool of the material which the termites structure and create underground, and this is left to differentiate. So the differentiation takes place inside this, and then once the mycelia which is ramifying this, it undergoes a particular stimuli. Probably there is definitely some sort of complex signaling which goes from environmental factors like change in rainfall, soil moisture and other things, because this part where the full pseudorhiza is covered, we have seen earlier also in the first part that was recorded with respect to this specimen, we have cleaned now the whole thick soil attachment from this part to this part, and here it is not clean intentionally, just to study the adherence of the clay particles, sticky clay particles to the pseudorhiza. And there are other specimens also which clearly indicate the role played by some kind of mucilage. So in the first part I provided some aspects of the various stages of differentiation of this root body, from the basal disc to pseudorhizal stage, from pseudorhizal stage to stipe stage, and from stipe to the full the epigeal developed mature stage of the Termitomyces mushroom. But the most interesting part of all this is, what is it that makes this clayish, reddish, lateritic soil stick to the surface of the pseudorhiza? Because we have done a lot of microscopic study and examination of the surface of the pseudorhiza, but one thing that we couldn't conclusively prove in the myco lab of Goa University Botany Department is what is the material that is coating this pseudorhiza. And it is possible because a lot of work has been done by Indian Institute of Chemical Biology, Kolkata, West Bengal, where they have done most of the work on a single Termitomyces species, that is Termitomyces clypeatus, which is known locally in Goa as Kut heimii or Kutiai heimii. because of the pointed umbonal region. And with the single Termitomyces clypeatus, they have published almost 100 papers, and they have also extensively worked on the Termitomyces clypeatus polysaccharide, exopolysaccharide. But that has come definitely from the pure culture. Here we are talking about exopolysaccharide production by the pseudorhizal surface. Now, what is the ecological and biochemical importance and significance of this type of exopolysaccharide secretion, and then isotropic coating, uniform coating in all parts that can be seen here, because this soil is almost permanently bound and forms a very complex polysaccharide plus clay complex. Means it is powerfully bound to the surface exopolysaccharide of the pseudorhiza, and then we find it extremely difficult to wash and remove this. We have to actually damage the whole pseudorhiza in the process of removing this. So sometimes the work is so cumbersome, so difficult, that the pseudorhiza becomes almost inseparable from the soil particles to which it is bound. Now, what is the practical importance of this is the binding strength of exopolysaccharide is so strong that we believe that the role that is played by the exopolysaccharide is to immobilize the abrasive nature of the soil particles through which it is transiting. So what happens is, from this point to this point, if there is a polysaccharide coating, we do not know what thickness, all along the length of the underground pseudorhiza, then we can see that polysaccharide acts like a biolubricant. So we are now propounding a hypothesis that Termitomyces life cycle is dependent upon the subterranean biolubricant production, and the biolubricant we also hypothesize is an unidentified, so far unknown exopolysaccharide, which is specific to this stage, that is the pseudorhizal stage from this point to this point, that is the basal disc to the emergence of style. So for this entire length we speculate, we hypothesize, and the proof, indirect proof at least, is provided by this inseparable. I am trying to remove this and it leaves a cast here. We can see the cast. The cast, if we examine very critically and microscopically, we will be able to see that a thin layer of the outer epicutis sticking to this, and that thin layer will be the coating provided by the biolubricant exopolysaccharide. So in this entire process, what happens is the transitional stage goes very smoothly through the application of the biolubricant. So in nature also we see that the exopolysaccharides come into picture in a variety of manner, and in this area I believe that the exopolysaccharide may also be having a lot of biochemical importance in terms of its action against undesirable lytic microorganisms, for which this is edible, this is nutrition. So this is a big question mark, because anything including seeds germinating in the soil, which comes through the soil teeming with microorganisms, and we are talking here about the fungus comb in an underground subterranean fungus garden, and it's not a sterile place, it is teeming with microbial life. There is a microbial community competing with the mycelium of Termitomyces inside the fungus comb and also in the vicinity. So this whole area shows that the style, as we can see, healthily fighting off all sorts of attacks, physical, biological and chemical. It survives, the pseudorhiza survives, and the battle scarred, the process of its survival through this stage to this stage is evidenced by melanization of the style. Now we have to look at the black spots here, melanized spots. This is definitely known as fungal melanin, and these spots clearly indicate the intensity of the reaction or the intensity of the friction or the intensity of the battle that the pseudorhiza has to wage or fight in order to complete its genetically programmed, the mission of coming above the ground and then completing this stage, so that the genetic commitment of this full species, this is a whole species Termitomyces, the full species is to form the reproductive organ. Those are very thick lamella, gills, and then billions of basidospores will be discharged on the ground, and they may be foraged back by the above ground termite workers, and the termite workers may re-inoculate fresh fungus comb with this vigorous new crop of basidospores, and the entire life cycle will be completed during the next season. That is fairly well known in Afro-Asian countries where Termitomyces phenology and biology has been well researched. But there are many small and big gaps left. We are trying to fulfill one gap by proposing certain hypotheses of pseudorhiza, biology. Because a lot of work is needed in understanding melanin biosynthesis in mutualistic termitophilic agaricales. How much is our knowledge? Because as far as the taxonomy is concerned, the taxonomist will point out to this dark umbonal region and they will say that this dark umbonal region is significant in differentiation and distinction of the species on basis of the length and the shape and the size and the thickness and all those aspects. But these are purely morphological descriptive aspects. What is the biochemical basis of melanization that we have to see? Now the standard explanation that will come out from any biochemist is this melanization takes place because of the emergence of the species out in the open, because this part is underground. It is not irradiated. There is no radiation. There is no ultraviolet. The part of the sunlight which may make it melanize, develop a black melanin pigment to protect itself, the soft tissue. But that fails because we see that the pseudorhizal stage itself here is very clear that it transits into this umbonal region. That means from the basal stage itself there is a genetic commitment, means there are definite set of genes which are involved in melanin biosynthetic pathway. Now if we try to understand the whole process of the pseudorhizogenesis of pseudorhiza from this point, that is primordia, attached to the fungus comb through a special organ called basal disc, to this point, we have different morphologies. This is one morphology, a longer pseudorhiza, slightly widening at the point of its differentiation into the stripe. We have a thinner, flexible, foldable type of pseudorhiza, means the mechanical aspects of the pseudorhiza also differ. This is hollowish. This is like I feel when I am touching this, the feel is like I am touching a tube, a plastic tubing, because I can see some air pockets inside. Now what is the meaning of those air pockets? What is that? What type of this vacuum is there or air is there inside? It is a true air or it is some sort of that condition that is created to give structural integrity to this type, because it is very well known in bioengineering or even civil engineering that as compared to hollow columns and solid columns or hollow tubes and solid tubes, the mechanical strength of a hollow tube or a hollow column is much greater than a solid tube or a solid column. So naturally, biomechanically speaking, the hollowness of the stripe definitely gives an ecological advantage to these Termitomyces species in that it doesn't break. But I can easily break the stripe and I can show that you can crack it. But it is very difficult here. It is very difficult because it is fibrous and hollow. Now I have cracked the stripe and you can see this. So this particular aspect is taken care of in the pseudorhizal stage, that this you can see the tissue structure is different. Its context is totally different. It is fibrous, filled, it is spongy, whereas this is flexible. So we have seen that structurally this is different, functionally it is different, and both structurally, functionally, this is the greatest miracle on earth that this species survives, and this has diversifies to give us 51 different species which are catalogued so far from many countries in Africa and Asia. And the beauty of the whole business is the biochemical complexity of melanin biosynthesis together with biolubricant synthesis of exopolysaccharide. Now when we couple both the events, then we have a very interesting model of ecological adaptation of the species, that these species can come above the ground and then form the healthy gills and discharge the basideospores, attracting lots of members of the local food web and food chain, which will find a very interesting nutrition here, because this is full of proteins, full of sugars, full of glycoproteins, full of carbohydrates and all, lichens. So this is a rich food source, and this food source will attract I think we have lots of beetles we can find inside this. I found that many of the holes which are created on this one are by the mycophilic, agarophilic beetles. So these agaricophilic beetles, I name them as agaricophilic beetles or termitophilic beetles or termitomycophilic beetle. That would be the right name. Means the certain groups of beetles which are termitomycophilic. Now my research student Dr. Miss Rosie D'Souza, who is cataloging this, recording this video, I will directly ask her the question whether you identify these beetles. What are they, which group they belong to?
So they are myco.
No, what is the group? Which lidae it is? Means what type of, close to which type of beetles? Please find out, because if you have forgotten the name, then we can record it later. But these beetles, this type of mushroom beetles have been extensively researched by Meredith Blackwell in Louisiana State University. Earlier, my student Chinchu Remnan, who did a MSc dissertation on mushroom beetles, she was interested in this type of beetles because we were interested in the yeast which are carried by these mushroom feeding beetles. They are having exclusive host-dependent association with these species. So my submission is this, that these beetles perhaps are specific to this mushroom. They may be around, but they simply means wait till these mushrooms emerge. So we are having now a more complex picture. Step one in this picture is that these Termitomyces species do not have independent existence anywhere on planet Earth. Second, at some point in the past, the Termitomyces species came to be associated with, the termites. Third point is the termites cultivated this in a asexual mode, and then you have the fruit bodies emerging in a very complex life cycle. Fourthly, after the mushroom emerges also, there is association of other faunal element. As you have seen that I have also recorded, and these video clips are available on YouTube, how gastropods, mollusks, that are snails at night, they devour, they graze on this. Understood. And local people say that primates and bears, they also consume this. Understood. We do not know much about birds, but as far as the insect fauna is concerned, we are quite sure that a large number of insect species, especially beetles, as we have seen them grazing on this. Now, this is as far as the apigeal stage is concerned, which comes into contact with all other members of the local food web. But as far as underground stage is concerned, there are many obstacles to be overcome, and those obstacles are overcome by the first, structural complexity of the pseudorhizal stage, two, the biochemical mechanism which is associated with the generation of the pseudorhiza, that is production of melanized region in the areas where it encounters a friction or a microbial attack or some sort of hard object, and then that whole part we can see that here it might have here it might have contacted a hard object or a piece of pebble. So the melanin coating gave it a very hard type of this one surface, and then it emerged further. But in the process, although we see lacerations and damages, it could successfully complete this stage. That means a battle is waged. This is the first instance of how living mushrooms wage a battle in nature. I think very little knowledge is there about the phenology of mushrooms in terms of what effort they have to make in the underground stage. So we are for the first time perhaps recording for genus Termitomyces. Anywhere in the world I can say that how much energy investment, resource investment this genus Termitomyces, species of this genus, have to make in terms of cellular currency, in terms of biochemical currency, in ensuring that this pseudoresal stage doesn't die its own death. Because if pseudorhiza dies, as far as all the apigeal fruit bodies of diverse Termitomyces species in Africa and Asia is concerned, this genus would be automatically extinct. So the submission in both the parts, the first part as well as of this whole narration and the second part of this narration is simple, that we cannot take the pseudoresal stage as just a taxonomic novelty or a taxonomic oddity or taxonomically unique. We have to go beyond that and we have to understand the biochemical basis and further go beyond that in future and understand the molecular basis, and I would rather say that we could go still deeper, and I feel that we could still deeper and understand the molecular ecology of pseudoresal genesis. So I would like to know your view about melanization while you are recording this video. What would you feel that if both the processes of the melanin biosynthesis and biolubricant exopolysaccharide synthesis are coupled, don't you think it is beneficial to the pseudoresal development?
Yes. Yes,
So
sir.
could you elaborate in what way you understood this whole phenomenon?
So it prevented from getting.
Like you see, this is very dark, and this is a middle part of the pseudorhiza. So definitely it has encountered a hard soil here, and then it has emerged.
Yeah, some rocks and pebbles.
Yeah,
also.
so that means aberration. Now, in terms of molecular ecology, how can we go deeper and have a hypothesis? Let us formulate a hypothesis like this, that this part of the entire pseudorhiza is blacker than the rest of the part. Can you see this?
Yes.
This is whiter, this is hyaline, this is almost colorless, and this is blacker. Then there are blacker spots here. This is black, this is melanized, this is melanized. Now let us talk about this part which is melanized. Now what are the triggers for melanization? Because the entire that tissue is mycelic. So there are specific genes which express this. Now it all depends on whether from the contact with the hard soil signals are communicated inside this tissue, so that specific genes involved in melanin biosynthesis are expressed. So our hypothesis is simple. We say that there are genes involved which are associated with external mechanical stimuli. You understood? Like what? That outer tissue of a pseudorhiza is encountering a hard obstacle. Now this information has to be translated into a biochemical signal, and that biochemical signal has to regulate expression of the genes which are involved in specific localized melanin biosynthesis, meaning the whole stripe is not getting black. So there is differential gene expression, localized gene expression in this area, like healing a wound, healing an injured tissue. Do you realize? So this process is poorly understood in terms of pseudoresal development. So I end the second video clip with this assumption that first and second part, when we take together, there is a long way to go before we understand all the intricate details of this very interesting, complex and beautiful genus Termitomyces. In the subsequent videos, we will try to shed some more lights on morphological, morphogenetic, taxonomic, biological aspects of Termitomyces species which we have collected from Goa University campus for purpose of global education, knowledge and research.